Hello, this is Kamcev from University of Michigan. In this video, I want to highlight a recent publication from our group on chloride-sulfate separations using concentration gradient-driven transport and anion exchange membranes. This article was recently published in the Journal of Membrane Science. You can get the article by scanning the QR code here, and the article is available open access, so free to read for everybody. This study is a follow-on to another recent publication from our group where we showed that concentration gradient-driven transport across cation exchange membrane or negatively charged membrane can yield exceptionally high lithium to magnesium selectivities. Here, if we take a standard diffusion cell and we put a cation exchange membrane separating the lithium-magnesium chloride mixture in a pure water compartment, and if we measure the concentration of cations in the pure water compartment, we find that lithium ions can transport readily across the membrane, but magnesium ions are largely rejected, thus giving us extremely high lithium to magnesium separation. The mechanism for this highly selective lithium-magnesium separation involves two different phenomena. One of them is selective ion partitioning within the membrane, and the other is uphill transport of the magnesium ions against their external concentration gradient. For the selective partitioning component, we found that chloride ions that enter the membrane are mostly balanced by lithium cations, whereas the divalent cations prefer to balance the fixed charges on the polymer backbone. Because concentration-driven transport is governed by movement of the co-ions, this means that lithium ions can selectively transport across the membrane along with these co-ions. We also found that uphill transport plays a very important role, and uphill transport occurs when you have a very low concentration of salt solution on the receiver side of the membrane. On this side of the membrane, the preference for magnesium is so strong at these dilute conditions such that the concentration gradient within the membrane can invert, and this leads to magnesium transport in the opposite direction. If you want more details on the study, you can click the link that's showing up on the screen right now. In this study, we wanted to see whether the same mechanism can yield highly selective anion separations. Specifically, we focus on chloride-sulfate separation because this separation is very important for applications like water desalination and direct lithium extraction. We synthesized a positively charged membrane or an anion exchange membrane that we call MOETMA GDMA, so this is a cross-linked membrane with a relatively high fixed charge concentration. And we tested both electric potential gradient and concentration gradient-driven transport of mixed sodium sulfate and sodium chloride solutions. In both cases, we actually found that the chloride ion moves faster than the sulfate ion, but in the case of concentration gradient-driven transport, the separation is substantially more effective. To investigate whether selective ion partitioning in these membranes contribute to the high selectivities that we observed, we performed competitive ion sorption experiments with chloride-sulfate mixtures over a broad range of chloride equivalent fractions in the solution. In these experiments, we also decoupled the counterions that balance the co-ions in the membrane, or the sodium ions that are mobile, and those that balance the fixed charge groups on the polymer backbone, or the quaternary ammonium cations. The partitioning results are shown in this equilibrium plot, where we see that the co-ions in the membrane are primarily balanced by chloride ions, and so the sodium ions that are governing the transport in this concentration-driven experiment are bringing along mostly chloride ions with them, which contributes to the high selectivity. We also found that the fixed charge groups on the polymer are also balanced by the chloride ions, which means that the polymer also likes to interact with chloride over sulfate selectively. We also tested the occurrence of uphill transport of sulfate in these membranes by modifying the diffusion experiment such that the donor cell contains a constant concentration of mixed sodium sulfate, sodium chloride solutions, about 0.1 molar each, and the receiver cell contains a variable concentration of an equimolar solution of both ions. In this plot, I'm showing the concentration in the receiver cell as a function of time for sulfate anions, and each curve represents a different concentration of sulfate, so 10 to the negative fourth to 10 to the negative 2. In both cases, we find that the concentration of sulfate in this receiver cell decreases as a function of time, which means that sulfate anions are moving against their external concentration gradient and diffusing from the receiver cell to to the feed cell or the donor cell. We also performed partitioning experiments where we mimicked each side of the membrane, so the side that faces the receiver cell and the side that faces the donor cell, and we measured the equilibrium concentrations of sulfate in the membrane. The dashed line represents the concentration of sulfate in the donor cell, which is a constant value, and the symbols represent the concentration of sulfate in the receiver cell. You can clearly see that the concentration in the receiver cell side is substantially higher than that of the donor cell, which means that the internal concentration gradient is inverted. Also, you notice that this concentration difference gets smaller as the total anion concentration in the solution increases, and you can also see that in the transport experiments where the flux or the apparent flux gets lower. Both of these experiments confirm that uphill transport of sulfate occurs in these membranes. We compared the chloride-sulfate transport selectivity of these anion exchange membranes to those of cation exchange membranes that have comparable chemical structures, fixed charge concentrations, and water contents. We did this because cation exchange membranes are also expected to exhibit chloride-sulfate transport selectivity on their concentration-driven transport on the basis of Donon exclusion. That is, negatively charged membranes should exclude the divalent anion sulfate more strongly than the monovalent anion chloride. In these experiments, we measured the total chloride and sulfate fluxes and normalized them by the concentrations of chloride and sulfate in the external solution. This gives us our selectivity. And we did these experiments with various solutions that have a constant total sodium concentration of 1 molar, and then we varied the equivalent fraction of chloride in the solution. The results are in this plot. For the anion exchange membranes, we could only measure a sulfate flux for the 0.1 chloride equivalent fraction in the solution. When we went to higher chloride equivalent fractions, the sulfate was below the detection limit of our instrument, and so the selectivities are presumably much higher than the one at 0.1 equivalent fraction. For the cation exchange membranes, as expected, we also saw chloride-sulfate selectivity, but the selectivity was constant across the entire solution composition range, and it was between 20 and 30. And so these results demonstrate that anion exchange membranes actually yield much higher selectivities than cation exchange membranes. We also measured the chloride-sulfate selectivity of various commercially available anion exchange membranes so that we can test the generality of the separation mechanism. We chose membranes that have different chemical structures, fixed charge concentrations, and water contents. The results, so here we're showing the separation factor of chloride and sulfate, which is the concentration of chloride and sulfate in the receiver cell normalized by the concentration in the donor cell, as a function of chloride concentration in the receiver cell. For all membranes, we find that the separation factor starts out at a really high value and then decreases as more chloride permeates in the receiver cell. The reason for this is that the concentration gradient of sulfate changes rapidly as the chloride concentration in the receiver cell increases, and this decreases the separation factor via this mechanism. What's interesting here is that all of the results collapsed on a single curve, and this indicates that the chemistry of the polymer or the water content and fixed charge concentrations are not very important for the separation factor. I'll end this presentation with some unanswered questions and also some interesting observation from this study, but also our previous study on lithium-magnesium separation using a similar mechanism. A key difference between cation separations and anion separations using this concentration-driven mechanism is the dependence of the separation factor on the type of membrane studied. For the cation separations, we found that the separation factor depended strongly on the type of membrane that we studied when we looked at several different commercially available membranes. Notably, we found that Nafion 212 exhibited extremely low selectivity, whereas membranes like the Fujifilm or the CR62 membrane exhibited orders of magnitude higher separation factor. One key difference between these membranes is their fixed charge concentration. However, the green data points, this is the CR61 membrane, actually has a similar fixed charge concentration to Nafion, so there's something else going on here that we don't really understand yet. If we look at the anion separations with the anion exchange membranes, all the commercial membranes that we tested, including our lab-made membrane, the results for the separation factor fall on a common curve. This means that the membrane type, or the chemistry, the fixed charge concentration, and the water content does not play a significant role in determining the separation factor. We need more studies with different types of chemistries and well-controlled systems to fully understand why there's a big difference between the cation separations and the anion separations. There's also a big difference in the way that cations and anions partition and distribute within these cation and anion exchange membranes. In both of these studies, we decoupled the two different populations of counterions in the membranes. Counterions that balance the co-ions and counterions that balance the fixed charge groups. In both cases, we found that the counterions that balance the co-ions are mainly the monovalent ions, so lithium in the cation exchange membranes and chloride in the anion exchange membranes. For the cation exchange membranes, we found that the fixed charge groups are largely balanced by magnesium ions, so the magnesium ions prefer to be near the polymer backbone. But for the anion exchange membranes, we found that even for the fixed charge groups, they're mostly balanced by chloride ions in the membrane. So this is a very big difference between monovalent and divalent ion partitioning in these membranes that point to specific ion effects that are not easily explained by their valence alone. We have quite a big effort in our lab in studying these types of effects, and so we're hoping in the near future we're going to be in a position to better understand these differences between cations and anions. Finally, a few comments on monovalent ion separations using this concentration-driven mechanism. In both of these studies, we tested monovalent separations, so lithium, sodium for the cation exchange membranes, and chloride nitrate for the anion exchange membranes. In both cases, we found essentially no separation between these monovalent ions. The selectivity for both cases was very close to 1. The reason for this is that there is not a significant difference in the partitioning between lithium and sodium or chloride and nitrate in these ion exchange membranes, and especially the partitioning does not depend on total salt concentration. If you remember, the uphill transport mechanism relies on having a very strong concentration dependence partitioning selectivity of one ion over the other. So for the monovalent ions, we don't see these effects, and so this mechanism does not work well for monovalent ion separations. We still have to test whether the mechanism works for other valencies that are higher than divalent, so for example trivalent or even tetravalent, but those studies remain part of future work.